alien DNA may have been found inside of humans, claiming it was inserted into our genes.
Something hidden in our DNA has surfaced, and what biologists claim about human DNA might turn the whole world upside down.
University of Wisconsin, Dr. John Hawks, did a comprehensive analysis of human DNA going back for many thousands of years, and what he found was astonishing.
Strange sequences are appearing where none should exist, raising a question no one expected to ask. If alien DNA was found, even as a possibility, what else has been hiding in the human genome all this time? The genetic anomaly. No one expected. The irregularity first appeared during a routine review of the 1,000 Genomes Project, one of the most widely used public data sets for studying human inheritance. A research team was checking parent-child trios when something unusual caught their attention. One child carried a stretch of DNA that did not match either parent. This wasn't a tiny mutation or a harmless blip. It was a structured piece of genetic code large enough to fall far outside normal variation. The team immediately suspected a technical issue. They reran the file through multiple alignment tools, checked server logs, and compared the sequence against archived reference genomes. Every test produced the same result. The segment stayed exactly where it should not be. Even the surrounding regions looked normal, which ruled out common problems like low coverage or compression errors. Inheritance mismatches do happen, but they almost always fall into familiar categories. Some appear as new mutations, others come from retroviral insertions or known structural variations passed quietly through generations. This sequence matched none of them. It did not resemble viral DNA. It did not match any known mobile genetic element. And it did not appear in standard variant databases used to classify unusual genetic activity. With no clear explanation, the team escalated their findings. They prepared the raw files and audit notes and sent everything to Dr. Max Rempel at the DNA Resonance Research Foundation. Rempel had a reputation for analyzing large data sets with unexplained non-parental markers, making him one of the few researchers qualified to review something this irregular. The internal researchers avoided speculation. They simply noted that the sequence did not behave like anything expected under Mendelian inheritance, and that its structure made it difficult to ignore. It was only one case, but it raised the possibility that similar mismatches might be hiding elsewhere in the dataset. By the time the file left the lab, the anomaly had already shifted from a routine flag to a problem none of them could solve. And because it came from a dataset used worldwide, they knew this unexplained sequence would not remain a quiet mystery for long. The expansion of the anomaly. When the irregular DNA sequence first landed on Dr. Max Rempel's desk, he didn't treat it as an isolated glitch. His work already involved searching through large datasets for unexplained inheritance patterns, so he returned to the same source where the first anomaly appeared, the 1,000 Genomes Project. This dataset includes hundreds of fully sequenced family trios and has long served as a foundation for population genetics research. Rempel decided to test the scope of the issue. Instead of reviewing scattered samples, he focused on 581 complete families, running them through independent variant checks to see whether the initial mismatch was a rare outlier or part of something larger. The results came back quickly, and they were hard to ignore. 11 families showed non-parental sequences, meaning the children carried genetic segments that did not match either biological parent. These weren't borderline variants or sequencing quirks that disappear under quality filters. They were clean, structured segments that stood out from ordinary de novo mutations. Across those families, Rempel logged 348 non-parental variants. The samples were all collected before 1990, which made one thing certain: none of the mismatches could be linked to modern gene editing. Techniques like CRISPR did not exist at the time, and no laboratory editing program had influenced public genetic datasets from that era. As Rempel went through each case, he noted something else unusual. The sequences didn't resemble viral DNA, which often leaves behind recognizable patterns. They didn't match mobile genetic elements, ancient hominin DNA, or known structural variants. Instead, the segments appeared unusually tidy. Their boundaries were clean, and their structure didn't reflect the irregular shapes produced by natural mutation or recombination. Even researchers who disagreed with Rempel later admitted that the patterns didn't fit easily into existing biological categories. After he finished reviewing the 1,000 Genomes project, Rempel expanded the search to a completely different group: people who self-identify as alien abductees. A few of these individuals had publicly shared their 23andMe data for independent analysis. Consumer genotyping does not sequence the full genome, so the resolution is limited. Even so, some families in this volunteer group displayed non-parental markers similar to the ones Rempel had found in the public dataset. This did not confirm anything unusual on its own. Array-based tools only read predefined positions in the genome and cannot detect new or complex insertions. What mattered was that the anomalies didn't appear confined to one dataset or one population. The same pattern surfaced in two unrelated sources, produced by different laboratories and different technologies. Media coverage picked up on that connection immediately. Reports highlighted that Rempel analyzed both population-level families and abductee volunteers. They also noted that his research came during a year marked by a spike in UFO sightings, which caused speculation to spread faster than the data itself. But the core findings were not based on stories or claims of contact. They were rooted in the inheritance mismatches that appeared on screen when Rempel reviewed the files. Still, Rempel insisted that the study's limitations mattered. He repeatedly emphasized that he could not reach firm conclusions with the datasets currently available. The 1,000 Genomes project was never intended to identify rare structural variants. The abductee data was even more constrained, since array-based platforms read only a small portion of the genome. In Rempel's view, next-generation sequencing or full whole genome sequencing was the only way to determine whether the anomalies were biological reality or technological artifacts. His caution did not stop the discussion from growing. Nigel Watson, author of Portraits of Alien Encounters Revisited, urged researchers to slow down. He reminded the public that abduction narratives can arise from sleep paralysis, psychological stress, or cultural influences. He did not dismiss further study, but he warned strongly against treating anecdotal experiences as genetic evidence. Even so, Watson agreed that deeper sequencing of volunteer abductees could resolve the question once and for all. If the anomalies were technical errors, the results would show it. If the mismatches persisted under high-resolution sequencing, then scientists would need to explain why. By the end of this stage, the debate was no longer about a single strange sample. Multiple families showed mismatches. The inheritance patterns did not fit what geneticists expect to see. The sequences themselves remained unexplained. And although Rempel's interpretation was controversial, the underlying data had opened a genuine scientific mystery, one that no existing model had been able to fully account for: the limits of current genetic tools. As Rempel's findings circulated among researchers, attention shifted away from what the anomalies might mean, and toward a more immediate question: could the existing data even detect these sequences correctly? The mismatches were visible, yes, but visibility alone didn't guarantee biological accuracy. To understand whether these segments were real or artifacts, scientists had to examine the tools used to find them. Most public genomic datasets, including the 1,000 Genomes project, were built using early-generation sequencing technology. These methods were excellent for identifying common variants shared across populations. They were not designed to catch rare, structurally complex segments. Older platforms often sacrifice fine detail in exchange for broad coverage, which means unusual regions can become blurred, compressed, or misinterpreted during the assembly process. This was the core of the scientific concern. If a dataset simplifies complex regions, it can make an irregular pattern appear more structured than it really is. A sequence might look like a clean insertion simply because the computational tools forced it into a neat shape. Consumer genotyping adds even more limitations. Platforms like 23andMe do not sequence DNA at all. Instead, they examine a fixed set of genetic markers selected for ancestry and medical reports. These tools cannot detect new insertions, cannot read structural variants, and cannot map unknown DNA. When Rempel noticed anomalies in the 23andMe data from abductee volunteers, he acknowledged these limitations openly. In his view, the array results were useful only as signals, not as proof. This is why Rempel publicly pushed for next-generation sequencing, NGS, and full whole genome sequencing, WGS. These technologies read DNA with far higher resolution. They can detect insertions, deletions, and rearrangements that earlier platforms completely miss. If the non-parental sequences were real, NGS would reveal their exact structure. If they were artifacts of older technology, NGS would expose that too. Some geneticists supported this approach. They agreed that unusual variants deserve deeper analysis and that the genome may contain rare segments that haven't been classified properly. But they also stressed that uncommon or puzzling sequences do not automatically imply something extraordinary. Human DNA contains thousands of rare mutations, many of which appear only once or twice in the entire dataset. These variants often look strange simply because they don't appear in reference panels. Other experts offered alternative explanations. Some pointed to the possibility of unknown archaic DNA. Denisovan ancestry, for example, went unnoticed until 2010, even though millions of people carried it. If another extinct hominin group left genetic traces in modern humans, the patterns might appear foreign, not because they were unnatural, but because science has not yet documented them. There were also technical explanations involving reference bias, a known issue in genomics. When sequencing tools encounter ambiguous regions, they often align the data to the closest known match in the reference genome. This can distort the true structure of rare sequences and create the illusion of an insertion or mismatch. But critics of these explanations focused on one detail: the structure of the sequences Rempel found. Natural variation tends to be messy. Over generations, recombination, mutation, and drift create irregular outlines. The segments Rempel documented were described as unusually clean, with sharp boundaries and repetitive features not commonly seen in human genetic variation. Even skeptics who disagreed with his interpretation admitted that the patterns did not resemble standard artifacts. Meanwhile, public discussion drifted far beyond the technical debate. Some people linked the anomalies to abduction narratives or claims of unusual abilities. Geneticists pushed back hard against this. They reminded audiences that sleep paralysis, trauma, and cultural influence can shape abduction memories, and that none of these experiences can be treated as genetic evidence. What remained was the central scientific question. If these mismatches were technological artifacts, why did they appear consistently across multiple families? And if they were biological in origin, why didn't they match any known category of mutation, virus, or archaic DNA? By this point, one thing had become clear. Whether the explanation was mundane or extraordinary, the current tools had reached their limit. Older datasets couldn't resolve the anomalies. Consumer platforms couldn't measure them. And until high-resolution sequencing was applied to these families, no one could say with certainty what the mismatches represented. The next phase of the investigation would depend entirely on that technology. Only deeper sequencing could show whether the genome held an overlooked chapter of human history, or whether the anomalies were simply artifacts that science had not yet learned to interpret correctly. The interpretation that turned everything upside down. Up to this point, Rempel kept his work firmly inside the boundaries of standard genomics. He talked about inheritance patterns, mismatched variants, dataset quality, and the desperate need for high-resolution sequencing. Nothing in his language suggested anything dramatic. But the moment he finished reviewing the 581 families from the 1,000 Genomes Project and the volunteer abductee data, he introduced an idea that changed the tone of the entire investigation. It was the first time anyone suggested a theory bold enough that some observers began saying that what biologists claimed about human DNA might turn the whole world upside down. Rempel argued that the strange sequences did not look like ordinary mutations at all. Their clean edges, their structure, and the organization of the non-parental variants seemed too deliberate to be explained by natural genetic drift. The 348 mismatched markers did not resemble retroviral activity. They did not appear in any ancient hominin reference database. They did not follow the messy, irregular pattern that recombination usually produces. To Rempel, the most reasonable explanation was that these sequences were not the product of human evolution as we currently understand it. In his words, humanity may be undergoing genetic transformation. This interpretation gained immediate attention because Rempel had not limited himself to academic datasets. He had also reviewed consumer-level DNA files from people who self-identify as alien abductees. Even with the limited resolution of array-based testing, a few of those families showed non-parental markers similar to what he saw in the public genomics database. Rempel said that if deeper sequencing confirmed the anomalies, it might even be possible to detect which individuals carry inserted sequences. That idea brought many viewers back to the same question. If alien DNA was found, even as a theoretical possibility, what would that mean for human identity? Rempel then expanded the speculation. He suggested that certain neurodivergent traits, like autism, ADHD, or Asperger's, might correlate with these insertions. He stressed that this was speculation and that no evidence supported a direct link, but he believed the idea deserved examination. He even suggested that unusual cognitive traits, such as heightened intuition or telepathic perception, could reflect genetic modifications that appeared long before modern science existed. These comments were enough to ignite controversy among geneticists and the neurodivergent community alike. His most debated statement came when he talked about long-term consequences. If hybridization with an external intelligence had occurred, he argued, humanity might eventually have to consider how much of that influence is healthy for the planet and which external sources should be given priority. Critics immediately seized on this. To them, the suggestion echoed fringe claims about unverified biological discoveries and raised ethical questions far beyond the scope of Rempel's dataset. Nigel Watson, who had already warned researchers not to jump ahead of the data, responded quickly. He reminded everyone that abduction stories cannot be used as biological evidence, and that extraordinary claims require extraordinary proof. Even so, Watson admitted one point. If high-depth sequencing eventually confirmed these inheritance anomalies in abductee volunteers, the results could be as world-shattering as recovering a physical craft. What made Rempel's interpretation so disruptive was not the narrative surrounding it. It was the fact that the underlying data still had no explanation. The mismatches existed. The structured sequences appeared in multiple families. The anomalies were not corrected or removed by any quality filter. They stood at the edge of current genetic knowledge, where the rules become unclear. Rempel's theory did not replace established science. It simply attempted to explain a pattern that did not fit any known mechanism. As media outlets amplified his comments, public interest surged. People who rejected the extraterrestrial angle still acknowledged that the anomalies represented a real scientific puzzle. And for the first time, the debate moved away from technical details and into a broader conversation about human origins, evolution, and whether hidden chapters of the genome are still waiting to be understood. Whether Rempel is correct or completely mistaken, his interpretation forced the world to confront a possibility that had never been seriously considered at this scale. And that possibility was enough to make many wonder if the claims surfacing from this research could eventually turn the whole world upside down. The shockwaves across science and society. Once Rempel's interpretation reached the public, the response inside the scientific world shifted instantly. Many geneticists had quietly reviewed the anomalies on their own, but now they felt obligated to speak up. It wasn't because they supported the extraterrestrial angle. It was because the conversation had moved beyond the data, and the loudest interpretations were starting to overshadow the actual findings. Their concern was simple. Real mismatches in inheritance patterns were being treated as proof of something far larger, even though the study had not been peer-reviewed and relied on datasets that were never designed for this level of interpretation. Research institutions reacted quickly. Labs began re-examining their own genomic archives, especially older trio studies used for medical genetics. Some revisited early sequencing projects that predated modern high-resolution methods. A few labs did find irregularities, but none were ready to interpret them the way Rempel did. For most, the anomalies raised technical questions about data handling, reference bias, and errors created by outdated genome assembly tools. Still, even the skeptics agreed on one thing. Rempel had drawn attention to a poorly explored corner of genomics, large structured variants that do not fit into existing categories. Government agencies quietly took notice as well. Departments involved in biotechnology oversight requested summaries of Rempel's findings and independent evaluations of his approach. Their interest wasn't focused on extraterrestrial claims. It was practical. If some people truly carried unexplained sequences, even if the cause was entirely terrestrial, it could change how genetic databases are curated. It could influence ancestry modeling, forensic identification, and the assumptions behind population-level studies. Unknown structural variants, no matter the origin, could disrupt the tools used across healthcare, law enforcement, and research. Outside scientific circles, public fascination exploded. The idea that what biologists claimed about human DNA might turn the whole world upside down spread rapidly across social media. The caution in the original reports was often ignored. Many people fixated on the possibility that alien DNA was found, even though Rempel repeatedly stated that he did not yet have the datasets needed for confirmation. Others leaned toward a more grounded explanation, seeing the anomalies as evidence of hidden evolutionary history. And as the debate grew, the line between speculation and data became increasingly blurry. Then came the reaction from the neurodivergent community. As one might expect, Rempel's suggestion that autism, ADHD, or Asperger's might correlate with these insertions drew immediate criticism. Many argued that such speculation risked creating stigma or misunderstanding. Geneticists supported this pushback, emphasizing that neurodevelopmental differences arise from complex biological and environmental factors, not from unverified ideas about external genetic influence. Rempel later clarified that he had presented the idea hypothetically, but the conversation had already expanded beyond his control. Nigel Watson's cautious approach gained momentum during this period. He reminded the public that experiences shaped by sleep paralysis, trauma, or cultural influence must be evaluated separately from genetics. Yet Watson also acknowledged one important point. If volunteer abductees agreed to undergo high-depth sequencing, the results, whatever they revealed, could provide real scientific value. A divide slowly emerged. Some people focused on Rempel's interpretation, while others focused purely on the anomaly itself. Even skeptics who dismissed the extraterrestrial angle still agreed that the mismatches deserved deeper investigation. The structured nature of the sequences remained unexplained. No consensus emerged on whether they reflected technical limitations, unknown ancestry, or a biological mechanism not yet recognized. By now, the fallout was larger than Rempel's theory. The anomalies forced scientists to reconsider how much of the human genome remains uncharted, simply because the tools used in earlier sequencing were not designed to detect rare or unusual patterns. Whether the sequences came from ancient hominins, unique biological events, or something far more controversial, the mystery was now in the open, and it demanded answers that current technology could not yet deliver. What comes next? With researchers split on the interpretation and public interest accelerating far beyond the original study, one thing is no longer disputed. The anomalies exist, and understanding them now depends on better data. Every scientist involved agrees that the current evidence is incomplete. Array-based genotyping cannot detect novel insertions, and older whole genome projects were never designed to identify rare structured variants with the precision needed here. If the unexplained segments Rempel highlighted are authentic, only high-depth whole genome sequencing can determine where they came from. Several academic groups are already preparing follow-up studies. Their aim is not to prove or disprove extraterrestrial involvement, but to test the inheritance mismatches using modern sequencing tools. These projects will require volunteers who are willing to undergo full genome sequencing and, in some cases, provide medical or family histories to help trace the irregular patterns. A small group of abductee volunteers has shown interest, though researchers emphasize that any personal accounts will remain separate from the genetic analysis. Across the field, one broader implication is already emerging. For years, rare or irregular sequences have been filtered out, dismissed as low quality, or set aside because they did not match existing reference models. Rempel's findings have forced researchers to re-examine those assumptions. The genome may contain more unclassified material than anyone realized, not because of outside influence, but because science has only recently gained the tools to study it in detail. Rempel still believes that humanity may be undergoing genetic transformation, though he repeats that his interpretation remains unproven. The controversy surrounds his theory, not the mismatches themselves. Those sequences are present in public datasets and are visible to any lab that looks for them. What happens next depends on deeper sequencing, rigorous collaboration, and a willingness to follow the data wherever it leads. Whether the explanation is an overlooked branch of human history, an unusual biological mechanism, or something far more unexpected, the investigation has already changed the questions scientists are willing to ask about our DNA. And the answers, once found, may reshape how we understand our past and our future. What do you think about this theory? Let us know in the comments. Thank you for watching, and see you in the next one.